Welcome to the learning module for nucleotide salvage pathways. This is Dr. Doug Tefke for Cellular and Molecular Medicine. After completing this learning module, students should be able to describe the overall fate of dietary nucleic acids, explain how free purines and pyrimidines are recycled in cells and identify the enzymes for this, identify the defect in purine salvage that leads to Lesch-Nyhan syndrome, describe the major symptoms of Lesch-Nyhan syndrome, describe why purine synthesis is activated or hyperstimulated in Lesch-Nyhan syndrome, and explain why Lesch-Nyhan syndrome patients have hyperuricemia and gout, and explain why allopurinol is an effective treatment to control their hyperuricemia. Dietary nucleic acids do not contribute energy as do carbohydrates, proteins, and fats. In the stomach, nucleoproteins are simply broken down by gastric acid and pepsin into nucleic acids and proteins. The nucleic acids, DNA and RNA, are then digested to nucleotides by the activity of pancreatic nucleases and phosphodiesterases in the small intestine. Nucleotides cannot pass through cell membranes. Nucleotidases and nonspecific phosphatases degrade mononucleotides into nucleosides, which can be absorbed through cell membranes. Once inside an intestinal mucosal cell, nucleosides can be further broken down by nucleosidases to free purine and pyrimidine bases plus ribose or deoxyribose. Purine bases are degraded to uric acid in the intestinal mucosal cells for secretion in the urine. Pyrimidines are degraded to water-soluble compounds in the liver. Most ingested nucleic acids are degraded and excreted. Therefore, nucleic acids are not a dietary requirement. Free purine and pyrimidine bases from the turnover of RNA and DNA and intermediates of synthesis are constantly released within cells. These free bases are in large part salvaged and reused to make nucleotides. Salvage consumes much less energy than de novo synthesis. It is the energetically preferred source of nucleotides for nucleic acid synthesis in almost all cells. The liver is the major site of de novo nucleoside synthesis and delivers free nucleosides and nucleobases to other tissues via the circulation, where they can be taken up and converted to nucleotides by salvage enzymes. Some tissues and organs, such as brain and bone marrow, almost exclusively obtain nucleotides via the salvage pathways. Free purine bases are recombined with phosphoribosyl pyrophosphate to form nucleotides. Two enzymes do this. Adenine phosphoribosyl transferase salvages adenine to form AMP. Hypoxanthine guanine phosphoribosyl transferase salvages hypoxanthine and guanine to IMP and GMP respectively. Defects in the enzyme that salvages hypoxanthine and guanine causes Lesch-Nyhan syndrome. The pyrimidines, uracil and orotate, are salvaged by orotate phosphoribosyl transferase, or OPRT, which is part of the UMP synthase enzyme used in de novo pyrimidine biosynthesis. Cytosine is first deaminated to uracil and then converted to UMP by the activity of OPRT. Thymine is not salvaged by recombining with phosphoribosyl pyrophosphate, as this would yield a ribothymidine. Instead, thymidine phosphorylase salvages thymine to the nucleoside thymidine. Thymidine kinase then converts thymidine to TMP, and TMP is converted to TTP by the activity of nucleoside kinases. Thymidine phosphorylase only uses deoxyribose 1-phosphate as a substrate for the salvage of thymine, so that only deoxy-TMP is made by this salvage pathway. It never utilizes ribose, This restricts thymine to DNA synthesis only. Lesch-Nyhan syndrome is caused by defects in hypoxanthine guanine phosphoribosyltransferase transferase, HGPRT, which is an X-linked disorder affecting mostly males. Hypoxanthine and guanine are not salvaged efficiently. IMP and GMP levels are reduced, while phosphoribosyl pyrophosphate levels accumulate within cells. Reduced utilization of PRPP for salvage means that PRPP accumulates, which can stimulate glutamine phosphoribosyl aminotransferase activity, hyperstimulating purine biosynthesis up to 200-fold. The excess purines that are synthesized are then degraded to uric acid. This causes hyperuricemia, excess uric acid in the urine. Often, orange urate crystals can be found in the diapers of affected infants. The hyperuricemia in Lesch-Nyhan syndrome causes severe gout in these patients, which can be treated with xanthine oxidase inhibitors such as allopurinol. For more information regarding hyperuricemia and gout, view the learning module on nucleotide catabolism. Lesch-Nyhan syndrome patients also display severe neurological defects, including severe mental retardation, tendencies for self-mutilation, biting of lips and fingers, and often have spastic involuntary movements. Here are a few questions to test your knowledge of nucleotide salvage pathways.